Hello, this is Miss Bichond again. We are going to take our notes on the genetic code. The genetic code. And when we're done with our notes, on this side of your notebooks, you're going to make a wonderful foldable explaining all the information in more detail. So here's the main point, is that your DNA has a code that gets translated into proteins, and everything that happens with you and how you look and how your body works has to do with the proteins that are made. So realizing that, the most important thing of all, as far as everything working correctly, are your genes. So what is a gene? A gene is a section of a DNA molecule that contains the information to code for one specific protein. To code for one specific protein. You remember that proteins are enzymes, proteins make up your muscles, proteins are hormones. Everything you need to live is mostly due to the proteins that you make. I am going to highlight the important vocabularies this time. So our gene there is a very important word. Okay, so how we use the word a section. How does one protein become human insulin and another section become the protein that makes up your growth hormone? Well, it has to do with the order. So I'm going to put a little asterisk here. It is the order of the nitrogen bases. Remember those? A and T and C and G. That's what changes in the DNA. The sides of the ladder, the sugar and the phosphorus, they stay the same. It's the bases that pair up. That order can be different, and that's the order that decides what kind of proteins are made. So it's these nitrogen bases, and let's just write them out here. We have guanine, cytosine, adenine, and thymine. Remember, A always goes with T and C always goes with G, right? And that's the order of those along a gene forms a genetic code that specifies, which means it tells, it specifies, it's telling a specific thing, specifies what type of protein will be produced. Will be produced. So I'm going to use a different highlighter for my nitrogen bases, and I'm just going to highlight. It's the order of the nitrogen bases. Always remember that, okay? All right, there's a new word coming up. We haven't talked about this yet. This is RNA. We've talked about DNA, but a new player comes RNA is ribonucleic acid. This little guy gets to leave the nucleus to go to the ribosomes for the proteins to be made. Remember, DNA does not leave the nucleus. So the RNA is like, you'll find out there's different types, but it's the messenger that takes the code from the nucleus to the ribosome. So, what is this? This is a genetic, we just call it a messenger. Which carries the genetic code from the DNA inside the nucleus. Remember, DNA is always in the nucleus. Outside, or we could just say into, the cytoplasm. We're so happy for RNA because without it, the DNA would have all this wonderful code for all these proteins to be made, and the cell wouldn't know how to do it because DNA can't leave the nucleus. So, again, this is a new vocabulary word. I'm going to highlight RNA. Now, there's a couple kinds of RNA, and one of them we call mRNA, and guess what the M stands for? It stands for messenger. Okay? So, mRNA is messenger RNA. That's easy to remember, right? And this RNA, what does it do? It copies the DNA's code, okay, and it carries it to the ribosome. Now, if you remember, ribosomes are where the proteins are made, okay? So it carries it to the ribosome. So mRNA is messenger RNA. But there's another kind of RNA, and we call that a tRNA. Not to be neglected, this one is also very important too. This is called the transfer RNA. Remember, to transfer something is to carry it or take it somewhere else. So I'm going to highlight the tRNA. What does transfer RNA do? Well, it does exactly what its word says. It carries. It carries the amino acids. If you remember, amino acids are one of our 4 biomolecules. They make up proteins, right? A bunch of amino acids together as a polymer make a protein. So if we're going to build proteins, we need the building blocks. That's amino acids. So the transfer RNA runs and fetches the amino acids necessary to build the protein. So you could think of it as a helper in a kitchen running to get the ingredients, okay? So it carries the amino acids to the ribosome and adds them to the growing protein. Adds them to the growing protein. Okay? So just to review so far, we have genes as a section of the DNA in the nucleus, but that does us no good if this can't get outside the nucleus to make a protein. So in comes RNA. We have messenger RNA, which gives the code to the ribosome, and we have transfer RNA, which carries the different amino acids needed according to the code, right? And then the proteins can get built. So let's do this. Let's make a little chart. Now that we've introduced RNA to you, let's make a chart that compares DNA to RNA. Okay? So I'm going to draw a little box here. I'm going to go 3 lines down. This is going to be a nice little graphic for you to understand the difference between the two. Now, I forgot to mention, but we always leave this for our Cornell notes. So if you wanted to, you could draw the line down here so that we can add our questions, sample test questions, and we'll leave a line down here for our conclusion. Like that. Okay, so what are we going to compare these two? We're going to compare, first of all, this stands for number, the number of strands. So if you remember, DNA has 2 strands, right? It's the double helix, like this. That's kind of a bad helix. But remember, they have 2 backbones. Well, RNA has only 1 strand. Only 1. Okay? The other thing that's different about them is the type of sugar. Y'all remember from the last quiz we had, the name of the sugar in DNA is the first letter of DNA, stands for deoxyribose. That's the 5-carbon sugar that makes up the backbone. Well, with RNA, it's different, and guess what? It's what the R stands for. It's just plain ribose. Okay? Ribose. Now, the other thing that's different are the bases. Okay? The nitrogen bases. For DNA, we have A and T, and we have C and G. Remember? The A goes with the T, and the C goes with the G. Well, when you get to RNA, there's one difference. And that is you have A and U for uracil. Uracil. That's the difference. And then you still have C and G. So I'm going to highlight uracil and the U. That's the big difference. Okay, 3 main differences. RNA has only 1 strand. Its sugar is ribose, and instead of T, we have U for uracil. So every time you see an A with RNA, it's going to go with U. Every time you see an A with DNA, it's going to go with T. Okay, there's one last thing that's very important, and that is, how does change occur over time? How do we have mutations? Well, it's just a change in the code. Remember, it's a section of DNA, a change in the code, and it's the nitrogen bases that make the difference. So if we just have a tiny change in those codes, that's how we get mutations. So let's say here, mutations. Okay, and again, that's another vocabulary word, so I'm going to highlight it yellow. Mutation. What is a mutation? A mutation is any change in a gene or chromosome. Remember, that's just the pattern of nitrogen bases. A gene or chromosome, which is a bunch of genes are on one chromosome. So that change will make a different protein, and that's where either a problem or a good change can come in. So let's say we're going to do an example. Let's say the original DNA says the red hat. Okay, There's 3 different changes that can happen. Okay, so do it like this. There's 3 different changes. One is called substitution. One is called deletion. Delete, right? Deletion, just like on the computer. And one is called addition. And you can figure out from those words right away, probably, the different kinds of mutations that can happen. So what if we substituted one letter, one little letter, and instead of an H there, we say the red cat. That's something completely different, right? A red cat or a red hat. So there you would have a mutation. What if something got deleted, and they always come in groups of 3? We'll get to that later. Always in groups of 3. What if we got rid of that D somehow? Well, then it would say the red, if we got rid of the D, the H would have to come over. So then it says the re at, and that's all messed up, right? Okay, so then what if we added something? And that can happen too. What if we added a letter? Well, then you have the read hat, and that's going to mess up the gene expression. So you have these 3 kind of mutations, and let's highlight those. You can have a substitution where you change a letter from one to another. You can have deletion where something gets cut off, and usually the groups of 3 shifts. Or you could have an addition, which they really don't like, and then you get 4, and that messes things up. So this can happen in this process of the RNA getting the code from the DNA and translating it into proteins at the ribosomes. Okay, I'm going to highlight ribosomes too. So you can remember it's the order of the nitrogen bases, and the proteins are made at the ribosomes. I'm going to highlight amino acids, and I'm going to highlight protein, because this is all about proteins. Okay, so there you have it. This is how the codes of your genes get translated into real life right here. DNA and RNA. There you go. So next is your foldable.